Imagine for a second that you are trying to rescue a message in a bottle. But, there's a catch.
Right, there's always a catch.
Exactly. The bottle is microscopic. I mean, it's made of this incredibly fragile, paper-thin glass, and it's currently just tossing around in the middle of a raging hurricane.
And your job is to somehow extract that bottle from the storm completely intact.
Yeah, so you can actually read the life-saving letter inside.
Which is terrifying, because if that glass cracks even a fraction of a millimeter, the message just dissolves into the violent water around it, gone forever.
Yeah.
You don't just lose the bottle, you lose the instructions inside that could basically change everything.
It really requires a level of precision that is almost hard to wrap your head around. So welcome to the deep dive. Today, we are going into the microscopic world of extracellular vesicles, specifically this fascinating subtype called exosomes.
Yeah, they are incredible.
They really are. And we are looking at a massive critical debate right now in the biotechnology industry, which is how do we actually harvest these fragile microscopic packages safely?
It's basically the ultimate engineering challenge in modern medicine right now. We're essentially comparing the traditional gold standard of extraction, which is this brute force method called ultracentrifugation, against a newer fluid dynamics approach called tangential flow filtration, or TFF for short.
Right. And the entire field is basically hinging on which method preserves the structural integrity of these vesicles better.
Exactly.
So we've got a fantastic stack of sources for you today. We're pulling from comprehensive scientific reviews that literally trace the 80-year history of vesicle research. We've also got detailed experimental studies on mesenchymal stem cell exosomes.
Which, for those who don't know, are essentially the specialized worker cells that help our bodies heal tissue.
Spot on. And finally, we're bringing in biotechnology industry papers focused on scaling these isolation processes up for actual clinical use. So our mission today is to figure out how these extraction methods compare when it comes to preserving the incredibly delicate membrane of these exosomes.
It's a huge topic.
Okay, let's unpack this. Before we can even judge the machinery used to extract them, we have to understand the cargo itself, right? Like, what exactly are we hunting for in that hurricane?
Well, to understand exosomes, you really have to look at how cells communicate with one another. For a long time, the prevailing wisdom was that cells mostly just secreted individual proteins or hormones directly into the bloodstream to send signals.
Like a single molecule floating through the blood to tell another cell what to do.
Right, exactly. But it turns out they also send out these highly complex bundled care packages.
Right.
Exosomes are incredibly tiny. We are talking nano-sized, typically between 30 and 150 nanometers in diameter.
Wow. To put that in perspective for you, a single strand of your hair is about 80,000 to 100,000 nanometers wide.
Yeah, it's wild.
So we are dealing with structures that are unimaginably small.
Unimaginably small. And they are formed through a very specific, highly regulated internal process called the endosomal pathway. Instead of just spitting proteins out one by one, the parent cell essentially swallows a piece of its own outer skin to create an internal pouch.
Wait, so the cell is basically pulling its membrane inward to make a pocket.
Precisely. It invaginates to create what's called an early endosome. And as this pocket matures deeper inside the cell, smaller vesicles begin to bud inward, packing into that original pouch.
Huh.
So the cell is meticulously packing these tiny bubbles with genetic instructions and specific worker proteins, creating a sort of biological shipping container holding dozens of smaller packages.
It's like blowing a bubble and then watching a bunch of even tinier bubbles inflate inside of it.
That is a great way to picture it. And when that large container, what we call the multivesicular body, travels to the edge of the cell and fuses with the outer plasma membrane, it releases all those tiny internal bubbles out into the extracellular space.
And once they're released into the wild, that's when we officially call them exosomes.
You got it.
What's wild to me, just reading the sources, is that they aren't just empty bubbles of fat. They are packed with a very specific, deliberate cargo.
Oh, absolutely. What's fascinating here is the sheer complexity of that cargo. Exosomes carry cytosolic proteins, which are the active worker proteins usually floating in the internal fluid of a cell, along with metabolic enzymes and crucial nucleic acids, including messenger RNA and microRNA.
So they are literally transferring genetic blueprints and functional machinery from a parent cell to a recipient cell.
Yes, and all of this is enclosed in a lipid bilayer.
Right. So going back to our analogy, these exosomes are basically microscopic water balloons carrying delicate, life-saving letters, and the rubber of the water balloon is that lipid bilayer.
That's
So
right.
if you pop that balloon during delivery, all the internal fluid, the proteins, the RNA, I mean, the entire message just leaks out into the void. The biological effect on whatever cell was supposed to receive that package is completely lost.
Which is exactly why the structure of that balloon is so vital. That lipid bilayer is highly specialized. It's enriched with specific fats, things like cholesterol and sphingomyelin.
Sphingomyelin, okay.
Yeah, you can think of sphingomyelin as a sort of molecular mortar. It forms these dense areas called lipid rafts, which give the exosome just enough structural stability to survive tumbling through the bloodstream while remaining flexible enough to eventually merge with a target cell.
But, stable is a very relative term at the nanoscale. Reading through the sources, the medical potential here is kind of mind-blowing.
Oh, it's revolutionary.
We are not just talking about research novelties. Biotechnology is looking at using these exosomes to accelerate diabetic wound healing, to modulate the immune system in autoimmune diseases, and even as targeted delivery trucks for cancer therapies.
But none of that happens if we pop the balloon while trying to catch it.
Exactly. To use them as medicines, we have to isolate them from highly complex biofluids without destroying that molecular mortar you mentioned.
And let me tell you, biofluids are an absolute mess. Blood, urine, or even the nutrient broth used to grow cells in a lab. These fluids are incredibly crowded,
Just full of junk.
packed with intact cells, fragmented dead cell debris, massive protein aggregates, other types of larger vesicles. Finding an intact exosome in there is like trying to find a specific, fragile glass marble in a landfill of jagged metal. You have to filter out all that junk to get to the pure exosomes.
So we have these incredibly fragile, life-saving water balloons floating in a crowded soup of cellular junk. To actually study them or use them as a medicine, we have to separate them out. But historically, the way scientists have done this is surprisingly violent.
Violent is definitely the word for it.
Yeah, we need to talk about the method that has been the undisputed heavyweight champion of exosome isolation for decades, which is ultracentrifugation.
Specifically, differential ultracentrifugation. If you look at the research published before 2015, roughly 81% of all exosome studies use this method.
81%.
Wow. Yeah, it was everywhere. It relies entirely on the basic physical principles of size and density. You take your complex biological fluid, put it in a tube, and subject it to a series of sequential spins in a centrifuge, increasing the speed and the gravitational force each time.
So we're essentially using gravity to separate the landfill.
Step by step, yes.
Mhm.
The first spin is relatively slow, maybe 300 times the force of gravity or 300 Gs for about 10 minutes. That pulls the heavy, intact living cells down to the bottom of the tube to form a solid pellet.
Okay, makes sense.
Then you take the liquid left on top, the supernatant, and you spin it again, maybe at 2,000 Gs to pull down the dead cells.
Right. So far, so good. We're clearing out the big chunks of debris without doing too much damage to the tiny stuff still floating in the liquid.
Right. Then you spin it at 10,000 Gs to get rid of the smaller cell debris and larger microvesicles, but to finally force those tiny, buoyant, 30 to 150 nanometer exosomes to sink to the bottom, you have to unleash massive gravitational forces.
How massive are we talking? We're talking about spinning the sample at 100,000 to 200,000 times the force of gravity.
You kidding?
No, and you have to hold the sample under that extreme force for anywhere from 70 minutes to several hours. The entire start-to-finish process can take up to 14 hours.
Okay, I have to jump in here and just push back on the scientific community a bit. We know that these exosomes are delicate lipid water balloons held together by microscopic mortar.
Yep.
If spinning these things at 100,000 times gravity literally crushes the very thing we were trying to study, why on earth did researchers spend 30 years using this as the gold standard? Are we just stubbornly attached to a machine because it's what researchers had sitting in their labs in 1987?
Well, it's a completely fair criticism, and the sources actually acknowledge this tension. Ultracentrifugation became the gold standard because it is conceptually simple and practically sterile.
Sterile, right. It doesn't require adding any external chemicals, salts, or precipitation polymers to the sample, which could contaminate the final medical product. It just uses physics. In the early days of vesicle research, nobody really grasped just how fragile these lipid bilayers were.
I guess that makes sense.
But you are entirely correct about the physical toll it takes. The sheer stress generated by 100,000 Gs is devastating to membrane integrity.
Yeah, the sources are pretty explicit about the damage. Under these extreme centrifugal forces, the exosome's lipid bilayer just ruptures. When that happens, the internal proteins and nucleic acids, the entire genetic message, just leach out into the surrounding fluid.
Furthermore, the sheer force mashes the exosomes together, causing vesicle aggregation. They are physically smashed into each other and into other contaminant proteins, forming these clumped, tangled masses.
Ugh, that sounds awful for the sample.
It really is. A clumped mass of ruptured vesicles no longer behaves like an individual functional exosome. It can't fuse with the target cell. Its biological function is effectively dead.
I read in one of the studies that the low-speed spins can even create outright artifacts, things called multi-layered microparticles.
It's exact.
We are talking about the mechanical force actually causing the lipid membranes to artificially fuse and fold in on themselves, creating these thick bilayered structures that do not exist in nature at all. So researchers using this method might not even be studying a natural exosome. They are studying a mangled high-gravity artifact created by the machine itself.
Which leads to an abysmal yield. The actual amount of intact exosomes you get back at the end of those 14 hours is terribly low.
Just brutal.
One of the comparative studies in our sources noted a recovery rate of just 29.13% using ultracentrifugation. Overall, industry estimates put the recovery anywhere between a meager 5% and 25%. You are destroying or throwing away the vast majority of your active ingredient.
Which is a massive bottleneck if you are trying to manufacture exosomes as a scalable medical therapy. You can't rely on a manufacturing process that annihilates 80% of your product and takes more than half a day to run a tiny laboratory batch.
Exactly. Knowing that ultracentrifugation acts like a high-speed blender to our delicate water balloons, engineers and biologists had to completely rethink the fluid dynamics of isolation. They needed to find a way to filter the junk without crushing the product.
Enter tangential flow filtration.
Exactly. This is where tangential flow filtration, or TFF, enters the picture.
So filtration seems like the obvious answer, but the sources make a huge point of differentiating this from standard filtration, because pushing fluid through a filter just destroys the exosomes in a different way, right?
Standard filtration, what engineers call dead-end filtration, has always been problematic for viscous biological fluids. In dead-end filtration, you apply pressure to push a fluid perpendicularly straight through a porous membrane.
Straight into the wall, basically.
Right. But biological fluids aren't water. They are thick, sticky sludges filled with proteins and cellular debris.
So standard filtration is basically like driving a massive truck straight into a tiny toll booth gate. The truck, which is our cellular debris, crashes into the gate and gets stuck. Suddenly, the millions of tiny cars behind it, our exosomes, can't get through at all. The whole system just clogs instantly.
That's a perfect analogy.
And because the pump is still pushing fluid into that clogged filter, the pressure spikes violently, crushing the exosomes against the trapped debris.
Yeah, in a fluid system, when that membrane fouls and the pressure spikes, the sheer force exerted on the exosomes squishes and pops them just as effectively as the centrifuge did.
Man, so I'm trying to picture the physics here. If pushing the fluid straight through the filter just crushes everything and spikes the pressure, the only other option is moving the fluid sideways. Is that what tangential means in this context? You're dragging the fluid across the surface instead of pushing it directly into the wall?
That is the brilliant pivot of TFF, also known as cross-flow filtration. The sample fluid doesn't get pushed directly into the filter membrane. Instead, a pump circulates the fluid parallel, or tangentially, across the surface of the nanoporous membrane.
Okay, so sticking with the traffic visual, TFF is like driving on a fast-moving, multi-lane highway alongside a very specific, highly selective exit ramp.
Exactly.
The traffic keeps flowing fast and parallel down the main highway, but the pores of the exit ramp are designed so that only the small cars, the exosomes, and smaller proteins can gracefully slip sideways off the highway and pass through. The massive trucks, the cells, and heavy debris just keep driving straight past the exit without crashing into anything. The sheer speed of the trucks zooming past actually continually sweeps the surface of the exit ramp clean.
That continuous sweeping action prevents the membrane from clogging. The heavy debris is constantly flowing across the membrane rather than being compressed into it. And because the exosomes are gently pulled through the pores by a slight pressure differential, rather than being subjected to the brutal compression of dead-end pressure or 100,000 G-forces, their flexible structures remain completely intact.
And the structural data from the sources is incredibly validating here. When researchers pull the exosomes isolated by TFF and look at them under a transmission electron microscope, they finally see pristine morphology.
Yeah, the TEM images show that classic cup-shaped lipid bilayer structure. They have a non-aggregated appearance, meaning they aren't clumped together like the centrifuge samples, and
Yeah.
they have very clear, distinct membrane structures without any artificial multi-layering.
They just look perfect.
They look exactly like they are supposed to look in nature.
But here's where it gets really interesting. The numbers back up the visuals. When we talk about yield, TFF is playing an entirely different game. In one of the specific studies isolating mesenchymal stem cell exosomes, the TFF method achieved an astonishing 91.88% recovery rate.
Wow.
Yeah, compare that to the 29.13% from ultracentrifugation. You are getting nearly all of your intact exosomes back, and the continuous flow process takes less than 3 hours instead of 14.
And, you know, it isn't just about the sheer quantity of the yield. It's about the biological quality of the cargo. Because the membrane integrity is preserved, the TFF-derived exosomes retain significantly more of their soluble factors. They hold on to their internal cytokines, their functional proteins, and their vital genetic material.
The message actually stays in the bottle.
And because of that, experimental studies show that these TFF exosomes exhibit dramatically greater immunomodulatory potency. They are functionally better at interacting with recipient cells and delivering their genetic instructions because they haven't been battered by extreme gravity.
It seems like a flawless victory for fluid dynamics. I mean, we get vastly more exosomes, they are in perfect physical shape, and they work significantly better biologically. And presumably, this is much easier to scale up for an actual pharmaceutical company.
Scalability is perhaps the most crucial factor for translating this research into actual therapies. If you want to manufacture GMP-compliant medicines, which means adhering to good manufacturing practice, you need systems that are entirely enclosed, easily sterilized, and capable of handling massive volumes of liquid.
Right.
You simply can't put 1,000 liters of cell culture medium into a centrifuge. The physics just don't scale. But you can easily hook up a 1,000-liter industrial bioreactor to a TFF system and just let the fluid continuously cycle across the membranes until the exosomes are fully extracted. TFF bridges that massive gap between small-scale laboratory discovery and large-scale pharmaceutical production.
But reading through the engineering sources, there is a catch. It is not a magical plug-and-play technology where you just pour the fluid in, turn the machine on, and walk away. The TFF machine itself doesn't automatically protect the exosomes, right? The specific parameters the operator sets do.
A vital distinction, yes. TFF gives you the capacity to be gentle, but you still have to actively manage the physics of the system. The most critical parameter you have to control is the transmembrane pressure, or TMP.
Oh, TMP, okay.
This is the pressure difference between the fluid flowing fast inside the main channel and the fluid that is passed through the membrane.
So what does this all mean? If we get these pressure parameters wrong, do we essentially just recreate the destruction of the ultracentrifuge just inside a fancier new machine?
Well, if we connect this to the bigger picture, yes, absolutely. If the TMP is set too high, you overpower the tangential flow. The suction pulls too hard, and you end up forcing the exosomes too aggressively against the pores anyway.
Right, creating that dead-end crash again.
Exactly. This causes membrane fouling, where proteins and vesicles get jammed in the filter, and it leads directly to the rupture of the exosome's lipid bilayer. The sources are very specific about this. They heavily recommend keeping the TMP low, generally under 15 CSI, and in some of the highly optimized stem cell studies, they restrict it to under 3.5 PSI to ensure maximum vesicle preservation.
You also have to tightly control the environment, right? I noticed the sources mention strictly maintaining the temperature at 4 degrees Celsius throughout the entire extraction.
Temperature control is crucial to minimize the enzymatic degradation of the exosomes. Even if you get the fluid dynamics right, biological material breaks down rapidly at room temperature.
I was also surprised to learn that even the physical shape of the filter membrane makes a massive difference in whether the exosomes survive. The sources specifically debate choosing between flat cassette packages and hollow fiber membrane columns.
Yeah, for delicate exosome purification, hollow fiber membranes are heavily favored over flat cassettes. A hollow fiber looks exactly like a microscopic straw. When fluid flows through thousands of these tiny, smooth straws, it creates a very uniform, laminar fluid dynamic that generates incredibly low shear stress at the wall.
Like a nice, smooth river.
Exactly.
Mhm.
Flat cassettes, on the other hand, can create turbulent, chaotic flow patterns that are significantly rougher on those flexible lipid membranes.
Yeah.
You basically have to consciously tune every single dial, the transmembrane pressure, the temperature, the microscopic geometry of the membrane, to successfully protect the product.
It's an incredible lesson for anyone involved in biotech or really scaling up any complex manufacturing process. The process is just as important as the product.
Oh, without a doubt.
You can have the most miraculous regenerative cellular therapy in the world, literally the absolute cure in a cell, but if your plumbing destroys it before it ever gets into the vial, it's completely useless.
The transition from ultracentrifugation to optimized tangential flow filtration represents a true maturation of the entire extracellular vesicle field. We are moving from the era of merely trying to prove that these microscopic vesicles exist to finally learning how to handle them with the incredible respect their complex biology demands.
Well said. To sum up this deep dive into the shifting landscape of biotechnology, ultracentrifugation was the blunt, heavy instrument that paved the way for discovering exosomes. It allowed researchers in the 80s and 90s to pull them out of the cellular soup and realize they weren't just cellular trash.
Yeah, got us started.
But as we look toward a future of next-generation therapeutics, tissue regeneration, immune modulation, cancer targeting, tangential flow filtration provides the gentle touch, the massive yield, and the industrial scalability needed to keep those delicate lipid bilayers intact and fully functional.
This raises an important question, though, and it brings up a truly mind-expanding idea hidden in the recent literature. As our isolation technologies have become gentler and more precise with systems like TFF, researchers have recently discovered the existence of things called exomeres and supermeres.
Wait, what are those? I don't think we've covered those yet.
They are non-membranous nanoparticles, incredibly rich in proteins and RNA, that coexist in the extracellular fluid alongside classic exosomes. They're even smaller and vastly more delicate,
Oh, wow.
which forces us to ask a rather provocative question. If our old blunt tools like ultracentrifugation were mashing everything together, creating artificial fuse layers and popping delicate vesicles at 100,000 times gravity, what other microscopic biological messengers have we been inadvertently destroying and ignoring for decades, simply because we didn't have a gentle enough filter to see them intact?
That is a staggering thought. I mean, how much of the microscopic world have we entirely missed just because our nets were too rough? It really makes you wonder what else is hiding in the hurricane, just waiting for a gentle enough hand to pull it out. Thank you for joining us on this deep dive. Keep questioning the process behind the science, and remember, sometimes it's not just about what you were looking for, but how gently you search for it. Catch you next time.